which we presented at CROI. The findings of it, since we had presented this we had discussed this at the International Drug Resistance meeting in South Africa last year, and the WHO recommended that we try to get this abstract in, which was in fact accepted as a theme discussion and a poster which Leonard presented last week. So he will share the findings of that study and then talk about the facilities of the Genomics lab, and we can see how it's a state-of-the-art lab, and I was very impressed with it in my visit last year. And I think it's very good that we all know about it for collaboration. So welcome, Leonard, both to the Department of Global Health, the University of Washington. I know it's your first time here.
Okay. Thanks, Dr. Barbara, and thanks, Stephanie, and thanks, Global Watch, for giving me a chance to present the study and also to showcase our facility so that it can be able to build additional collaboration that we will be able to work together. So today I want to present a surveillance study that we did where we assessed the frequency of the dolutegravir resistance among ART experienced viremic patients in Kenya who are receiving DTG-based ART. The study was done jointly under the leadership of NASCO, but supported by the Genomics and Molecular Surveillance Lab, which sits at the National Public Health Institute, and also the Mosaic, the PEFA, and the USAID team. Next. So I would first of all like to give a background on the dolutegravir resistance. So WHO recommended that patients in resource-limited setting be switched to DTG-based, and the reason was because of the high genetic barrier for the insulin regimen. And in that specific recommendation, they are given that all patients to be switched, regardless as to whether it's their first initiation or their treatment experience, and that was basically because of the high success rate for high treatment success rate because of the tolerability of the DTG-based regimen and the efficacy, and then also the relatively high genetic barrier for the second generation of insulin, that is the dolutegravir, and cabotegravir, and there's low risk of resistance in treatment for naive patients who are starting with DMD and other DTG-based regimens. So it's based on that aspect that WHO gave recommendation for people to be for PLHIV to be transition. Next. The implementation of that, as Dr. Gad indicated, presented in CROI 2024, and also in one of the HIV meetings in South Africa, was that there was blind transition to DTG-based regimen, irrespective of whether the patients were viremic or they were non-viremic, and there was also transitioning to DTG-based regimen for both naive and treatment experienced patients, and because of the increased time on DTG-based regimen and limited value monitoring that happens in some of the resource-limited settings, the breakdown in terms of testing, there's been a margin report of a high proportion of DTG drug resistance in some of the observation and lab-based studies that have been done. And that was reported first by Dr. Gad in South Africa, but also in the CROI 2024 meeting. Next. Again, if you look at the IS resistant list, it indicates the mutations that come up, especially for patients who will be initiated with DTG. One of it is G118R, G140, and Q148, and then R263. So the three are some of the mutations that Dr. Gad highlighted in CROI 2024 as one of the possible mutations that usually come when somebody is initiated on a DTG-based regimen. Next. So it's based on that background that we decided to assess the frequency of these mutations for patients who are were viremic within Kenya. So we assessed the frequency of the DTG-based mutations. Next. So to provide the context of Kenya, so Kenya rolled out DTG in 2017 as both first and second line of treatment for PLHIV. So it's based on that we decided to assess the frequency of DTG using CADA surveillance. The expected sample size was 622 from the western region of Kenya. The sample was supposed to be collected between January and March 2023 to examine the frequency. Out of that, we also got the demographic and treatment data from the National HIV Viral Program. And then after that, we performed genotypic testing for all samples that had viral above 200 copies per ml. It's also good to provide the context that Western Kenya is approximately, the approximate prevalence of HIV in Kenya is approximately 4%, translating to 1.36 PLHIV, million PLHIV in Kenya were on ART, and out of that, 1.28 million are currently on DTG-based ART. Relatively, it's good to note that the average prevalence of HIV in Western Kenya is 11.3% across the 9 counties, if you consider an approximate, and that actually holds 73.8% of the Kenya's HIV burden. The 9 counties in Western Kenya consist of counties in Nyanza and also the former Western province. So that's based on the HIV estimates 2020. So out of the expected sample size of 620, we managed to get 190 within the surveillance by the time when we were presenting this data. Out of the 190, 41 were ART naive, and as you can see, like on the green side, shows the ART naive, and then on the pink one shows the ART experience. So we have 149 who are ART experienced. By saying ART experience, we mean that they were initially on an NNRTI or PI-based ART before they were switched to DTG. By saying that they're ART naive, it means that DTG ART was their first line of treatment. The median duration for the patients who are ART naive on ART was 1.5 years, but the median duration for the patients who are ART experienced on DTG was 1.1 years.
Yeah, that's it. That was the.
Yeah, next.
Oh, no. Okay, yeah, that's the one.
Okay, so this table presents the demographic details and the demographic and details for the ART naive and ART experience. As you can see again on your right, on the green one, we show the median age. The median age for the ART naive and also the ART experience. And there was no significant difference in the median age. There was no significant difference, especially in the female stratification, as to whether they are female or male. However, there was significant difference, especially the time that they've spent on ART. As you can see, the ART naive is 1.5 years, but the cumulative time spent on ART regimen for the ART experience was 7.5 years. The time on ART prior to DTG or prior to DTG ART, which is 5.2 years for the ART experience. And then the time on DTG, the time that they spent on DTG is 1.5, and then 2.1, which was also not significant. The viral load before switch to DTG for the ART experience was 90, was low. 62% of them had no viral load before switch to DTG. The one that viral load is below 200 copies per ml is 17, and the viral load above 200 copies per ml is 30. So that's for the ART experience. So for the DTG naive, they were initiated on DTG, and there's no baseline viral load in Kenya, so we don't expect the viral. Next. So this figure shows that out of the 190 samples that we got, 41, out of the 41 ART naive, 13 out of 41 were having viral load above 200 copies per ml, and 42 out of 149 were having viral load above 200 copies per ml. Out of the 13 samples that of the ART naive, we managed to sequence 12 out of 13, and then out of the 42 samples we managed to sequence of the ART experience, we managed to sequence 31. One among the 12 had an INSTI mutation, and then 7 out of the 31 had INSTI-DR mutation, translating to 22.6% among the ART experience. So that shows the higher INSTI-HIV-DR resistance among the ART experience compared to the ART naive individual. There was a 70% had resuppressed viral load below 200 copies across the two groups. So the resuppression was more or less similar across the two groups. And this table summarizes the INSTI mutations, the ART experience of the 8 individuals, 7 among the ART experience and 1 among the ART in the naive. It shows the previous regimen before the DTG based regimen. It shows the durations that they had been on ART. It shows also the viral copies by the time the sequencing was being done, and it shows the major INSTI drug resistant mutations. You can see E138K is common among all of them, and then it shows the interpretation over what those mutations mean, as whether it's potentially low level, it's high level, it's intermediate or susceptible. So as you can see, we have 5 high level resistance among the 7. 1 is intermediate, and then 1 has potential low level. While for the naive, it's intermediate. The R263K has also been indicated in the ICE list as among some of the mutations that comes out from some of these initiatives. But you can also see that all the 8 had got that had the INSTI-DR mutation, or also had reverse transcripted drug resistant mutations, and across all of them, and that could be one of the main reasons. And this the reason has also been highlighted within a number of studies about the existence of these DRMs and terms in fueling up the rising DTG mutations. So in summary, if we compare the ART naive and the ART experience, there was no difference in age, gender, and approximately 70% had suppressed viral load on DTG-based ART in 2 groups. The ART naive had a shorter duration on DTG, that's 1.5 years compared to the ART experience. Among the ART experience, we had 7 out of 31 translating to approximately 23% of had HIV INSTI resistance, and 5 conferred high-level resistance to DTG. So that's actually higher if you compare that to the ART naive that is 8%, 1 out of 12 that had INSTI-DRM, which was actually an intermediate resistance to DTG. And all the 8 with INSTI mutation have an additional NNRTI mutation, and that, I think, indicates the need, especially to be able to have a very strong drug resistance surveillance program and also viral program to ensure that these issues can actually be identified. And if we have to preserve DTG as our first line of treatment. Next. Yeah, so just the conclusion that the frequency of INSTI drug resistance was high among the viremic ART compared to the ART naive. And this data suggests the risk of developing instant drug resistance mutation among those with background of pre-existing mutations, such as the NNRGI. And the study again emphasizes the importance of early and HIV monitoring in HIV-based on DTG-based ART. One of the limitations of the study was that it was a small sample size and convenience, and as such, there's need to determine DTG drug resistant mutation at a larger level. And I'm happy that MOH through NASCOP and NPH has taken up this, and they're intending to conduct ADR surveillance across the country that has got a much more robust sample size of 1,400. So we believe that data will be able to add much more and to shed much more light on this specific data set. Yeah, I would like to thank the WACh study team, Dr. Chauhan, Dr. Woodby, Perry Pedros, for supporting this study, both technically and also supporting financially and with resources. My colleagues in the lab, Vera, Rukia, Peter, and Dr. Kiru, who supported the study. The NASCOP team, we have Dr. Rosa Fuller, the current head NASCOP, who been very supportive, and we also gave in her technical input on how she wanted the data to share. Dr. Caroline Mwangi, the healthcare treatment NASCOP, who also been supportive. Dr. James Wagude, who had the NYWES. The NYWES is where the samples were coming from. It's a technical working group that manages the treatment and the care of the HIV, people living with HIV. And then Dr. Mambo, who was former healthcare treatment lead in NASCOP, but has now moved to WHO. I would also like to appreciate my institution, that's the NPHI, the Ministry of Health in general, NASCOP, the WACh study team that has been supporting the study, the USAID and the PEFA team that support the viral program and the technical aspect of drug resistance surveillance. And the study participants who also gave in their samples. The study wouldn't have been possible without the study participants.
We can take questions if anybody has any questions at this time before Leonard shares the facilities for the lab.
Yeah, I would probably wait for questions and comments, and then after that, be able to move to the next presenter.
Yes, please feel free to come off mute or enter a question in the chat box. Yes, Irene.
Hi, thanks, Leonard, for the presentation. Sorry, I couldn't be there in person, but I'm just curious for the, so was the total population, the 190 patients, all of them at the time of the sample collection, was it based on the fact that they were not virally suppressed? And then when you repeated the testing at the HIV reference lab, like, were some suppressed? I just for clarification. Very nice presentation.
Yeah, so initially these patients were supposed to come from the viremic clinic, assuming that they were not virally suppressed based on their last viral load. But now when we repeated the test, we found that some of them had suppressed below 200 copies per minute.
So was there like some time duration between when the initial viral load testing had been done at the facility and the time you got? Because I'm curious, is this based on remnant samples, or this was fresh sample collections that were done for purposes of the study?
It's fresh sample collection. So
okay.
Within the viremic clinic, they have patients. So they call them to be able to participate in this surveillance. And then, so we had to confirm again the VL. So if we are doing a remnant-based survey, we don't repeat viral load, but for this specific case, we had to repeat viral load because the fresh sample. Most of them is between 6 to 3 months viral load.
Okay.
Yeah, from the time that the last viral load.
Okay, thanks.
Yes, Irene.
Thanks so much, Leonard, for that presentation. I was also wondering, in terms of the recruitment for the people who are in the viremic clinic, do you have that data of, who's the population that we got these samples from? Because what I noticed is that it's quite a young population, and so your median age was 30 and 22. And when you look at the ones who are ART experienced, it seems like it's mostly perinatally infected because they'd been on ART for a long time. And I just wanted to understand whether, there were people whether, like, for example, the Mosaic study had some eligibility criteria that would have led us to get predominantly young people, or whether that's actually the population that was there.
I think it's common, especially within the majority of the patients within the viremic clinic are actually young, and I think that has been able to help design the current ADR survey in Kenya, because initially, if you see the WHO guidance, they recommend the assessing of DR between 19 and below and 20 and above. But the Kenyan protocol has been modified to also to get specifically the ADR prevalence for between 15 and 19 and 19 and 24. So the two subcategories have actually been informed by the type of population within the viremic clinic. So I think the patients the PLHIV that participated in the study just depict the current population within the viremic clinic. So, and majority of the patients within the viremic clinic also falls within that age category. Most of them are relatively young.
But just to comment, I know we are seeing a lot of DTG drug resistance. It's quite shocking. The numbers are very small. We were wondering whether this data should be, so we did seek permission from NASCOP in the treatment and care before we could even send in the abstract to CROI. But they encouraged us that we should probably start thinking right now and do a larger sample size, as he was mentioning it in his conclusion. But a drug resistance to DTG at 22.6 seems like very high, in the population, especially now that 90% of our population is on TLD, the adult population, and kids are on Abacavir-based DTG. So it's a little scary as well,
But also to add that we have a good suppression rate of approximately.
It is about 70%, yeah. But some at CROI were mentioning that they are seeing 90% suppression, in other countries. So what are we, DTG has been longer from the other resource-limited settings in Kenya, which started in 2017. So we could have had longer exposure. Some of them were mentioning they had the DTG start at 2019. So we'll have to see what we come out with the result from a larger sample size, to look at DTG drug resistance. Yeah, we need to closely monitor. We need to do more viral loads and drug resistance testing. And I know resources are very limited, even in purchasing the test reagents. It's very expensive to do drug resistance as well. And there was a debate in NASCOP about whether we should be, if one has a failure, and it's viremic, you counsel the participant, and then you ask them to come again after, is it another 3 months? You see viral loads are still high, you counsel, and then by the time that come the third time, it's now you request to NASCOP. That was the old guidelines. And I remember NASCOP TWG treatment and care was debating whether now that DTG has got high genetic barrier and it's very efficacious, should we, since we don't have funds and to buy reagents should we stop doing drug resistance. But this data is showing the other way around, so we need to really think and present this data at the National Program Treatment and Care and see what they think about getting resources for constant viral load monitoring and then also doing drug resistance. So I think it's an important data. Mm-hmm.
Yes, Irene, you have another question or comment?
No, it's not a question. It's actually a comment about what Bhavna is saying, because I think it's difficult to make a conclusion at this point. And I agree that we definitely need more data. But I think also if you look at the slide, and I forget, I don't know whether you're able to put back on the slides, because if you look at the previous regimen that the patients were on, I think that's really like an important beginning point, because the treatment experience, and I think maybe point of clarification, when we refer to treatment experienced patients, are we referring to patients who are previously on ART by primarily first line, or previously on ART and had gone through a cycle of several regimens, maybe they were on second line, third line, and I think that's going to be a useful distinction, for example, to make, and it appears like many of them were actually on potentially what could have been their first line, like within their NRTI and NRTI-based first line. So given that the country did, and I think it's not only Kenya, countries did this massive transition of patients from NNRTI-based first line to DTG, I think that's an important context to put in place. But I definitely agree that we need more data to be able to make any substantive conclusion. The Nyanza Western region also has a lot of activity. We don't know what's happening. Like, it's one of probably the very active regions. So we don't know what's happening in the other regions, but it would be useful to interpret this based on just a much larger sample size, and also to contextualize based on all the transitions that have happened previously in the country. These don't appear like they were patients who were actually on second line. They look like they were patients. Okay, some of them were. So it would be good to get enough numbers to sort of make the distinction.
Yeah, in agreement, yes.
Any final questions before we move on to the next presentation?
Oh, sorry, it might be more of a comment, but adding to that conversation, it might be helpful to also understand among women during pregnancy, and if some of the transmissions that we're seeing are also amongst women that maybe have some of these mutations that are highly acceptable for resistance, in terms of that being a target population also that might be helpful for, if we think about ministry and trying to gather more data to help inform policy. And if the resources are limited, there might be high-risk groups that could be targeted specifically, and that might also be one of those populations. Over.
Yes, I am in agreement. I think it's very important, isn't it, Leonard, to look at pregnant women, especially now that DTG is the main treatment which is being given. So if a child if the mother has mutations and transmits it to the kid, and the child is infected, vertical transmission, we need to know all that because usually drug resistance isn't done at the time of start of treatment, isn't it? It's not a policy. Christine will keep that in mind when Leonard is talking to NASCOP and making decisions about the surveillance. It's a good population to include. Yeah.
Okay, so I'm going to present about the National Genomic and Molecular Surveillance Lab, which was initially within the National Public Health Lab. The transition has seen some of the labs moved now to the National Public Health Institute. So next. And it's one of the labs that have been moved to the National Public Health Institute. So the National Public Health Institute is newly created. It has 5 directorates. The lab falls within the Directorate of Public Health Laboratory Science and Safety. Within that, we have 5 divisions. One for specialized public health laboratories that kind of gets up all the reference labs to be able to fall within that specific division, especially the service reference lab that provide quality material for diagnostics, but also coordinates the entire aspect. So I know of interest, a lab such as the HIV reference lab will end up falling within that specific division. Then we have Division of Genomics and Bioinformatics, which is now being used by National Genomics and Medical Surveillance Lab. Then we have a Division of Quality Assurance, Control and Standards that will be able to encompass all the aspects of the QA material. Then we have the Division of Biosafety and Biosecurity that will majorly be concerned with the border protection, but also the biorepository of samples and also the safety of staff and the samples. So within the Genomic and Molecular Surveillance Laboratory, we have 5 staff, 2 staff who are on fellowship, 2 implementing partner staff, also within the division. It's located within the KNH compound, National Public Health Lab Block B for people who know the place. It has 5 rooms, 1 dedicated for clean room, 1 room dedicated for extraction, 1 for PCR processes and library preparation, 1 room dedicated for sequencing, the core sequencing center, and then 1 also dedicated for sequence quality assurance. With the priority areas that it's interested in, one is for the AMR surveillance, whereby we apply pathogen genomics for to get detailed mechanism of the resistance within the organism, the transmission pattern, the enabling aspect of the resistance, and basically to enable the healthcare practitioner and policy to implement targeted infection control for the various MR and also in terms of the outbreak. The second one is to support emerging infection and biosecurity, whereby we do it to serve as the early warning system for emerging infection. I think probably if I'll be able to get a chance to present again, we'll be able to present on the national dashboard for genome pathogens. And then the third is on the vaccine preventable disease and elimination program, whereby we do pathogen genomics to support vaccine preventable disease monitoring, support identification, interruption of disease, transmission chain, look at the ones that are achieving elimination targets such as malaria, and also to ultimately contribute to improve assessing of vaccine efficacy and also to enhance population. So the ongoing work that's there, we have the routine HIV/DR testing. The annual volume in 2023 was 2,343. We had some reagent shortage in 2023. In 2024, we are expecting reagent, so we hope to continue. SARS-CoV-2 sequencing, that's an ongoing process. 674 complete genome sequence, most of them are from wastewater within the last quarter of last year and then this year. HIV/ADR surveillance that we expect to support from mid-2024. It has a sample size of 1,886. That's the one that are indicated. It's going to support to verify the data that we have found. The TB targeted NGS. So with the additional molecules for the TB patients, the lab is expected to support in TB NGS sequencing as we transition from culture-based to targeted NGS. Approximate sample size is 6,000 to 7,000 on an annual basis, but we are also going to support the TB ADRS survey, where we'll be able to support them for whole genome sequencing for the sample for the TB ADRS survey. Cholera sequencing, where we've done the 267 isolates. We've shared 2 policy briefs with the ministry concerning the current cholera outbreak. The E. coli sequencing, among the dysentery cases, 368 isolates. We've completed the sequencing and we are working on the policy brief. Klebsiella pneumonia sequencing, where we are currently supporting 39 isolates from 3 clinics in Kenya. HPV/HIV co-infection studies. Those are planning studies, especially from the previous FIA that we are working on, the HPV sequencing, the HCV phylogenetic study, the wastewater-based surveillance, the protocol which we are still ongoing, and then also the metagenomics, where we try to resolve the cases for the course of certain outbreaks in Kenya. Next. Still continuing, we also support the stability study verification and validation, Candida aureus sequencing that we expect to start, and the expected sample size is 375 in October this year for one of the studies. The viral surveillance protocol being supported by Global Fund, the E. coli, and then also the influenza sequencing. So most of this work have been supported by Global Fund, but also MOH through the FRAC project. Next. So what have we done before? We've been able to support the OLA simple validation assay. I know most people here. Okay. So OLA is a study that is a point of care that we were trying to we were identifying probes to be used for doing point of care drug resistance. So we were able to support that specific study. It's being supported by NATADA and Professor Barry's team. The OPT4 studies, where we are supporting the HIV-DR component, that's being funded by University of Pittsburgh. The DTG outcome study being supported through CDC, the Institute validation study, MOH, the CADA HIV surveillance, again, MOH through Global Fund, NASCOP, and then the SARS-CoV-2 sequencing during the pandemic, supported through Global Fund, and then the Klebsiella pneumonia for being supported by APHL and also Global. Next. So what equipment do we have in the lab to support this? In terms of extraction, we have Kingfisher Flex, Epigenics extractor, and then the Abbott 2000. For PCR, we have the thermocyclers, the 8 thermocycler, and then we also have a Quant Studio and 5 and a Quant Studio 7. So Quant Studio 7 allow us to do Taqman array analysis. We also have for automated library preparation, where most of the mistake in the sequencing work happens, especially due to pipetting work, but we have a 57 automated for liquid handling and also for the library prep. For the sequencing, we have the API 3730 Sanger, which we majorly use for HIV, the ion tolerant PGM that we also use for HIV, DR, but also for HCV and HPV. We have the nanopore mini-ion MK1C and 1B. We majorly want to transition that platform, especially to our mobile lab for support outbreak investigation. We have Illumina MySec, and then we also have a NextSec 1000 that supports the bulk of the sequencing. And then we also got a nanopore GridION. So both the last two are high throughput equipment in terms of the sequencing. To assess and assure the quality of the sequence, we have a tape station that enables us to look at the sizes of the fragments that we generate if they are as per the standards, the qubit, and also the GelDoc that we continually assess at various steps of the sequencing path. So I would also like to share about the routine pathogenomic surveillance being supported by Global Fund, where we are trying to assess the certain diseases and to be able to identify them on time. So we have 20 sentinel sites across the MOH facility. It's across all ages, gender, and then all groups. In that, we are getting the 10th to 12th patient, where we get the different sample type. It's based on volunteering and based on a verbal consent. It's being spearheaded by the disease surveillance program, but supported by the lab. We get different sample type based on the WHO definition criteria of what sample type to get based on the symptom. So we have the 10th sample where we get the plasma and the swab, the 11th sample where we get the stool swab, and then we have the 12th sample where we get the oropharyngeal swab. So these facilities are located across the country, and we get the sampling on a monthly basis, and so that we are able to proactively identify an outbreak before it actually occurs. So it's expected that on the lab side, once we identify upper pathogen, we are running them on a viral surveillance panel. Some of them we also do on metagenomics. If we identify upper pathogen, we are supposed to share that sequence length with Cambridge. We are now supposed to develop up now primers to enable an intensive identification of that specific pathogen within that specific facility using the open PCR platform that were input in the country during COVID. Next. So this just give a pathways of the various work that we are going to do under that specific work. So we have the respiratory plot, the enteric plot, and then also the AMR plot. For the respiratory plot, we have the client comes, the collection, the transport, extraction that we do. We run them on the viral surveillance panel. We do the bioinformatics using base space and Dragon. Once we identify, we report to MOH, but also share the sequence length with Cambridge. The Cambridge now develop customized diagnostic kits for intensive identification of that particular pathogen within a specific location. On the 16 samples, we get the stool and the rectal swab. That's the path it goes. Again, it goes undergoes the same path. So initially, the oligosynthesizer was supposed to be done by either INRI or Cambridge, but right now they put it in Cambridge. We're now supposed to assemble up the diagnostic kits. The MR board also moved through the same way, where we sequence, but once we sequence, for the MR, we majorly share them as policy brief, but we also do it as part of the quality assurance to the Fleming Fund project. So this is an intensive work, and it's being supported by Global Fund. We have 3 years to complete it. For the Fleming Fund side, we are supposed to support close to 6,000 whole genome within the 3 years to be able to do quality assurance for the AMR work within the country.
So you do respiratory diseases, enteric, and also acquired microbial resistance.
Yes. So this is the dashboard that probably next time I'll be able to share the dashboard. I think my team shared with me the dashboard today in the morning, but it was so late to be able to incorporate it.
We can invite you, to give us
So yeah, I'll be able to share just specifically now on the dashboard. Next. So the menu within the lab. So again, we do HIV drug resistance. We do molecular network surveillance, where we do genetics. That's for the HIV. For the TB, we also do TB drug resistance to support case support. And also for the various surveys, we do transmission molecular analysis. For malaria, we support malaria drug resistance, where we are looking at the drug resistance to the drug that they're using, the resistance to diagnostics, where we are interested in the HRP2, 3G deletion, the resistance to vector and insecticide, and then elimination surveillance. The back of malaria surveillance, I must say, it's currently being done at Cambridge Wellcome Trust, but we are in the process of now transitioning now to be done at the GMSL. For the AMR, we are still continuing with AMR, the Klebsiella, the cholera, the E. coli, but also now covering up the entire scope of the 10 class pathogens for surveillance. And then also for the outbreak surveillance and management, we do metagenomics, and then we have various data expression. We majorly use CZ, that's the Chan Zuckerberg for the data expression, and then also we do R for data expression to look at, because we have to look at the most abundant, especially for metagenomics, to identify the correct cause of the specific pathogen that's responsible for the outbreak. For the vaccine efficacy, it's still pending validation, but we are working with Cambridge to ensure that we transfer up the support to the GMS. Next. So these are the pathogen genomic surveillance sites where we do the surveillance. It's not covering all counties, but it covers the entire country, and it gives a picture of what happened in the entire country. So this just to ensure that the country is in a good pandemic response posture in case there's a need. Next. We did a joint mapping exercise with CDC to look at the potential aspect of growing up the lab, the areas of collaboration, potential collaboration, why there's need to be able to centralize up genomic sequencing in one lab as a means of sustaining up the sequencing, the training up of staff that's required for both the wet lab and the wet lab processes, and then also how to better support the sentinel sites to ensure that we actually achieve the expectation of MOH and APHI. Next. So that's it. These are some of the lab equipment that are in the lab and the lab team that's currently supporting the work. Thank you.
It's very impressive.
So I'll leave this slide up because it's really neat. Okay, so let's open it up for questions or comments.
Can I ask you a question? So you are doing a lot of next generation and whole genome sequencing, and do you have facilities to do all the bioinformatic analysis and things like that?
Yeah, we currently do bioinformatics in-house. So a number of staff have gone for training, but we also have the aspect of transferring up various pipelines from Chemwell. We work very closely with Chemwell Wellcome Trust and then also Association of Public Health Laboratories. Yeah,
FPHLS.
Yes, FPHL, to be able to transfer some of the pipelines, especially when we start up a project. We usually work on the bioinformatic pipeline that's required, so they're able to install the pipeline. We have close to 700 terabyte of storage space of the genome sequencing, and then it also has got a high computing environment, and that courtesy of global.
So your staff are trained well to do that. They've had training.
Yeah, the staff have had training, but usually in bioinformatics, there's what we call service bioinformatician. So service bioinformatician is whereby you already have a pipeline, and then
they help you out.
Yeah, you train yourself on that specific pipeline, and then you just implement. But then also they are coding bioinformatician. So we don't have coding bioinformatician. We say that most of the time we are like service bioinformatician, but we would be interested much more to learn, especially on the aspect of coding, to be able to develop our new solutions, especially challenges that comes.
Is there something you mentioned about metagenomics and some tunes or something?
Yeah, Charles Zuckerberg. So it's just like what I'm saying about the service bioinformatician. So Charles Zuckerberg has a platform whereby once you've done your sequencing, you put up your sequence data and it does for your expression, then you can actually be able to assess and do a report on the potential cause of that. Yeah. So there are two aspects of doing it. There's actually an analysis using SQL, but then also analysis using R. So both we've used.
We have a comment from Grace. Impressive lab capacity and great presentation.
Thanks, Grace.
It's nice to know that we have the state-of-art lab which can support and they're willing to collaborate, open to collaboration. They've supported NATADA's study as well. And things which we need to do, whole genome sequencing or bioinformatics, metagenomics, we can probably do this in country now with this kind of facilities. And they do have a system of payment, isn't it? Like if we are doing a collaboration and we want to do payment, so they do have costings for per test. So if we want to, we can just collaborate, write up a collaboration, and we can pay for doing the storage of samples as well. They have a lot of facility of -80 freezers. How many do you have? I've seen your repository.
Yeah,
it's a big room.
We have 38 -80 freezers, but we are currently installing liquid nitrogen.
I'm quiet.
We are currently installing just liquid nitrogen to help storage all this.
So you do have liquid nitrogen facilities as well?
Yeah, it's currently being supported and again by Global Fund under the COVID-19 fund. So we got some funding to support just liquid nitrogen storage.
Do you have cryosuits or you have tanks?
Tanks, it's going to be
Okay.
tanks. Yeah. Tanks with also the specific virus for study. Yeah.
And I know that they have all the software for keeping chain of custody of samples and things like that.
Yeah, we have the
I've been very impressed with the lab.
It's really great food for thought. Those on the call, if you are interested in working with the lab, yeah, please feel free to reach out to Bhavna and Leonard.
Christine has raised her hand.
Yes, Christine.
Sorry, I keep raising it at the very end. Yeah, just a question. So it looks like this is fantastic in terms of the lab infrastructure and really exciting. So thanks, Bhavna, for asking those questions, because it's exactly what I was thinking in terms of how could we work together and potentially, instead of shipping things out, have some of the microbiome work done. So Leonard, has your team worked on any, it sounds like they probably have, in terms of the shotgun sequencing or 16S and for the microbiome, the gut microbiome work? And would that be something that they have the capacity to be involved in here? And then lastly, I missed it, I think. Is this lab in Nairobi or is it in Kilifi? I believe I heard Kemri welcome, but I'm just wondering where this infrastructure is currently located. Thank you.
I'll start with the first one. For shotgun sequencing, we haven't done, but we have the capability of doing that, and we'll be willing to support that specific aspect. The lab is located in Nairobi. Kemri Welcome Trust is in Kilifi, but we work in close collaboration. They've done a lot of staff training at our lab, and for most of the training, they've actually come to our lab and does the training within our lab as part of the capacity building for the lab staff for both the wet lab and the bioinformatics processes. So the lab is located in Nairobi.
Nairobi, just in Kenyatta National Hospital compound at the back, Christine.
Oh, perfect. Okay, great. Thank you. All right, so it sounds like the equipment's there. So just maybe you haven't yet started on kind of the microbiome, but that could be an area that we could work together and try to build a capacity.
Yes.
Awesome. Thank you.
In fact, Leonard is very interested in doing his postdoc in microbiome. He just finished his PhD, so we have been trying to approach if there's a possibility he can do a placement somewhere, maybe 3 months, and learn some microbiome work. Since they do have equipments and facilities, they could probably start up all the microbiome work there in Kenya. Looking forward to that, Leonard.
Sure.
Yeah, let me know, Bhavna. I think we could talk about that. I'm sure you already have many other people mentioning that they probably have other possibilities, but that placement, I wonder if we could help with that. If not me per se, but someone else on one of our studies that we're all intimately involved in, that would be really great if we could help facilitate that.
Yeah, I was going to approach Heather Jasmine, and we'll see while he's here to have a meeting there with her.
Yeah. And I've seen Irene's comment. Thanks, Dr. Irene. I must acknowledge Irene and Sarah were initially the people created out this unit.
Really? Thank you, Irene.
And even the first aspect of staff. So thank you, Dr. Irene. I hope this is a dream that you people have.
And you're leading.
Yeah, no, it's really impressive. I think to see the development from the time when there was nothing and a lot of push and pull to get equipment into the lab. So really, congratulations to the team. It's super impressive. So keep it up.
Mm-hmm.